All right, hello grade 10s. We are going to finish off chapter 7 here by talking about animal and plant cells. So here's a few did you knows for you about cells. In general, a person contains about 100 trillion cells. And a lot of those are our friends, the red blood cells, right? There's actually 200 different cell types in mammals like us. For instance, here we have a picture of a neuron, which is going to look a whole lot different than a red blood cell. Cells are tiny. They're very small. They measure about 0.002 centimeters or 20 micrometers across. So 1,250 cells shoulder to shoulder would be about an inch, or here in Canada, we would say 2.2 centimeters or a couple of centimeters. All right, so I know you did some cell stuff in middle school. So here's what a typically idealized animal cell could look like. Of course, we have our nucleus here and a nucleolus and an endoplasmic reticulum, something called the cytoskeleton, which we're not going to talk a whole lot about, but like those are like protein tubules that just help the cell maintain its shape, and a bunch of other things. We have, of course, the mitochondria, and we're going to get into each one of these organelles, which is what we call them, in our cytoplasm or cytosol. We're going to take a look at each one individually and talk about its function. But you should be able to recognize it in the context of your whole cell diagram. Near the end of this PowerPoint, there's lots of blank slides you can practice on where we've got arrows pointing to unlabeled cell diagrams. So you need to know what things look like, as well as know their function. Guys, this is very much a structure function kind of situation. All right, so let's deal with the fact that we often think of the cell analogous to a factory or to a city. Again, in the video clip section of my classroom, you will have lots of videos there that you can watch to give you more information about this. So again, if everything was analogous to a factory or a city, we would have the control center, which is the nucleus itself, right? It's going to control and direct the activities of the cell. We've got a bit of an assembly line because factories make something. So cells are usually in the job of making something. Often it's a protein, and that takes place in the endoplasmic reticulum, which has our workers, the ribosomes, as part of it, and we would call that rough endoplasmic reticulum. We also have a distribution center, sort of a Amazon warehouse, so to speak, where things get packaged up, refined, and processed, and then packaged and sent out. So that's the Golgi apparatus, Golgi body. We have the cell membrane, which is going to act as a control for what comes in and what goes out of the cell. We have our lysosomes, which are there sort of as security and also as a recycling kind of center. The mighty mitochondria, of course, the powerhouse of the cell. Everybody knows this from high school. What else do we have? I think I've mentioned pretty much everything we have here. So let's start dealing with each one of these things individually. Here's our nucleus. Now the nucleus has pores. And so sometimes it's called the nuclear envelope. It has a membrane and it has pores so that things can go in and out. And that's one way that the nucleus is able to communicate with the rest of the cell. It's also the area where the nucleolus is found, which is a dark, dense region inside the nucleus. It's thought that that's where the ribosomes are sort of made, kind of like the workers' staff room, so to speak. All right, and when you go further on in biology, you learn all about transcription and translation, which is the protein making or protein synthesis step. And then we're going to get into things like the DNA, of course, is housed in the nucleus. Those are the instructions that the cell can follow, but the DNA never leaves the nucleus. If the cell wants to get something done, it has to send some RNA, a cousin to DNA, which is single-stranded, which carries information, and that can leave through those pores in the nuclear membrane. And those are the instructions that can be followed and carried out by the ribosomes in the endoplasmic reticulum. So speaking of the endoplasmic reticulum, it's where protein synthesis occurs. About half of the cell's proteins can be made here. Ribosomes, of course, we said, often dot the endoplasmic reticulum. They build the proteins. And it allows for protein movement through sort of the passageway. So it's, again, it's likened to an assembly line. Now the lysosome, we mentioned sort of like a recycling security center. You can consider it a digestive bag of enzymes. So essentially, it's going to greet something that might come into the cell, friend or foe, and then it can break down whatever that happens to be. And those particles can maybe be used to be recycled, or maybe those particles will have to be expelled from the cell. Okay. By the way, the lysosome is not found in plant cells. This is an animal cell thing. Now, to our mitochondria. Now, mitochondria is double membraned. It has this oval-like structure. The inner membrane is all wavy and forms structures called cristae. And when we see wavy, curvy things in nature, what does that usually tell us? It's all about surface area. So that's going to increase surface area for chemical reactions that happen inside the mitochondria. Powerhouse of the cell. So its job is to provide energy. How does it do that? Well, it takes a hold of glucose molecules and it oxidizes them. So there's a reaction with oxygen that happens inside the mitochondria. We talk lots about this in grade 11. And that reaction is called cellular respiration. And so glucose plus oxygen, the products will be carbon dioxide and water. And the whole goal is to make cellular energy, which is known as adenosine triphosphate, or ATP. Now, both plants and animal cells will contain many mitochondria, depending on the energy needs of the cell. So, for example, a sperm cell, which is going to be in constant motion, would need many, many mitochondria. Now, the Golgi apparatus, which is usually a collection of Golgi bodies, looks kind of like a soggy stack of pancakes. Its job is to collect often proteins that have been worked on by the endoplasmic reticulum, package them, and distribute them. So there's some synthesis there, but there's also some packaging. And we call the finished package that leaves the Golgi a vesicle. So vesicles are like small containers that are going to be used for transport inside, or maybe ultimately to fuse with the cell membrane and deposit materials outside the cell. Good. Okay, now we're going to look at an idealized plant cell. Quite often plant cells don't have, they have more of a rectangular structure, but notice there's going to be three major structures that are different between a plant cell and an animal cell. And that's going to be a cell wall. Okay, so we're going to have a cell wall. Now we still have a cell membrane. All cells have cell membranes, but plant cells have a very rigid, stiff cell wall. And we'll talk about that in a second. It also has a very large central vacuole. You can find vacuoles, which are really just storage containers inside animal cells, but it's very pronounced inside of a plant cell, because a lot of what it's going to store there is water, and plants need lots of water. So it gives them that ability. And then we're also going to see chloroplasts. So chloroplasts are plastids that, again, give the plant cells the ability to convert light to sugar. So we know that plants are autotrophs. They can feed themselves. So the chloroplast is the organelle that makes that possible. So there's the chloroplast, and it has stacks of discs inside it. And again, this time, it's the discs that create the surface area for chemical reactions to take place, all for turning light and the energy of photons of light into glucose, which is sugar that can then be stored and, remember, converted into ATP, cellular energy, by the mitochondria when needed. All right? Again, it's double membraned as well, just like the mitochondria. Solar cell is a very good analogy for this. Again, if we think of a factory, okay? Okay, just a reminder that in order to do photosynthesis, we need water, carbon dioxide, and then the chloroplast is going to be able to convert that into sugar and oxygen as a byproduct, which we can be very thankful for. All right, and besides that, we have most of the other structures that animal cells have. But there's the three: cell wall, a central vacuole, and a chloroplast. These slides are again just kind of going into the details of exactly already something we have already said. Guys, you don't have to worry about the details here of the cell wall. Primary walls, a lamella, secondary walls, that you don't have to worry about that. That's not tested material. However, it is, like, to make it strong, layers. Layers make it strong. And quite often it's composed of a structure called cellulose. Like I said, at the end of the slideshow, we have some blanks to test your knowledge. If you were to get a diagram, which you probably will in your test, you're going to get blank animal cells and blank plant cells with arrows and numbers, and you should be able to match structures with and functions. Okay, there's our plant cell. There we go, we have a legend. And that's the end of our show today. Short lesson, guys. Happy studying.